Hello, thank you very much for joining the YouTube channel of Advanced Multiple Incorporation. Advanced Multiple Incorporation actually is knowledge processing and information management organization located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with the Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Both of these organizations are located in Mississauga, Ontario, Canada. Okay, I'm going to discuss today about principle and tools of gene therapy. The principle or the tools or the instrument, devices, protocol, procedure, techniques used in gene therapy. For the presentation today, I collected the information and scientific data from different sources, including PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, US, MBAS, then Duage, Directory of Open Access Journals, ProQuiz, CNKI, that is China-based database company that provide the scientific research information all over the world. Some of the information collected from the Crossref and ResearchGate. Before moving forward, I would like to discuss a little bit about fundamental things, basic stuff, gene technology, principle, and scope. It is the branch of biological sciences which deal with study and technique used for insertion or removal of the gene from living organism to produce desired quantity in them, the desired quality or the desired quantity of the proteins within the body. In fact, it is a process of transferring specific gene from the chromosome of a gene organism and transplanting them into the chromosome of another organism in such a way that they become a reproductive part of the organism. The method of changing inherited characters of organism by altering its genetic material is known as genetic engineering. Genetic material contain information for development, organization, and the function of living system. So, gene therapy is the insertion, alteration, or removal of the gene within an individual cell and biological tissues for the treatment of certain monogenic disorder, genetic disorder that involve to restore the normal functionality of the body, organ, or the cell. So, genetic engineering developed in the medical science. That is a very important discipline and a very important subject in the field of the medical science, in agriculture, in health science, dentistry, everywhere. Development of the sophisticated tools improve safety and efficacy. Highly efficient gene targeting strategy and site-directed gene editing technology developed after approximately more than 1900 clinical trials. The gene therapy then moved from vision to the clinical reality. It became a reality from the dream. So gene therapy revived as a powerful treatment option for the correction of the monogenic disorder. It can potentially be used for the treatment of cancer, blood disorder, blindness, AIDS, cystic fibrosis, muscular dystrophy, Huntington disease, COVID-19, hemophilia, and Parkinson's disease. Okay, well, there are the certain tools, certain component or the segment of this discipline for insertion of the human gene into the bacterial cell for the synthesis of the gene product, which replicate highly useful substances are employed as tool of the genetic engineering. The most important tool or most important component or the equipment used for the genetic engineering are enzyme, vector, and the complement DNA clone or complement DNA library. So there, there are the three fundamental segments or three fundamental components of the genetic engineering that are enzyme, vector, and the cDNA or the cDNA library. Well, we are going to discuss the first segment or first component of this discipline, that is enzyme. Enzyme used as microbial tool to in recombinant DNA technology or major enzymes are maybe the restriction endonuclease that cleave or the break the DNA double strand into the single strand DNA. Restricted endonuclease are two main type. The type 1 is endonuclease. Recognize the specific sequence of the nucleotide on the DNA. They cleave only one strand of the DNA. Type 2 endonuclease recognize a particular target sequence in the double-strand DNA. This gives rise to the DNA fragment of the defined length and sequence. Number two is nuclease that split the nucleus. DNA ligase, that is also an important enzyme for bacteria and tissues and the cell. We know very well there are these certain drugs like ciprofloxacin, ofloxacin. These drugs actually stop the function of the ligase, DNA ligase. This is a very important enzyme in the bacteria that help in reproduction, replication, and reparation of the bacterial DNA. When we inhibit this enzyme, it means we have inhibited the replication, reparation, and the reproduction of the bacteria. So eventually, the bacteria will be died, and we can control the bacterial infection. So the DNA ligase is one of the type of the enzyme. Alkaline phosphatase, then a reverse transcriptase that converts RNA into the DNA, then DNA polymerase, then to produce the copies or the clone of the DNA. Vector. The vector is the second component of the genetic engineering or gene editing. Vector are the DNA molecules that carry the DNA fragments. Viral vector used for the human gene therapy are retrovirus, adenovirus, adeno-associated virus, lentivirus, herpesvirus, then herpes simplex virus. These are the examples of the viruses that are frequently used in the research, academic, and the businesses to transfer the gene from one DNA to the other DNA. Viral vectors are three main types, that may be the plasmid, maybe the bacteriophage, maybe the cosmid. A plasmid is a small circular double-stranded DNA molecule that is distinct from a cell chromosomal DNA. Bacteriophage is a type of the virus that infect bacteria. Cosmid, a type of the hybrid plasma contained a lambda phage cos sequence and used as a clone vector and building the genomic library. Non-viral vectors, there are the 6-7 different kinds of the non-viral vectors that are frequently used for the transfer or the delivery of the genetic material. Injection of the naked DNA, electroporation, gene gun, sonoporation, use of the oligonucleotide, lipoplexies, polyplexes, and inorganic nanoparticles. Well, if we talk about the cDNA, like complement DNA clone bank or the complement DNA libraries, a cDNA clone bank or the cDNA library is the complement DNA produced from the messenger RNA. A type of the characterized cDNA molecule is inserted into a suitable vector and later transformed into the bacterial cell. The transformed bacterial cell containing plasmid of the cDNA is called a cDNA clone. This is actually the scope and principle of the gene therapy. There are the two main type of the gene therapy used all over the world. Germline therapy is the transfer of the gene to the sperm to the egg cell. Second is somatic cell gene therapy, involve the transfer of the gene to the cell. There are the two main approach used in the gene therapy: the replacement or the replacing a mutated gene with a normal regular gene. Inactivation or the knocking out a mutated gene that is abnormal, that is producing the problem, that is not functioning properly, that is knocked out, inactivated. Third one is the addition of the new genetic material into the body to help fight against the disease. So there are the three main approach that is successfully used in the research in the academia or in the business laboratories for the purpose of the gene therapy. In the diagram, it actually highlights the principle of the gene therapy. The therapeutic transgene, therapeutic mean medical or the pharmaceutical, transgene mean the particular segment or the part of the DNA that is delivered into the cell, attached at the particular receptor or point at the cell membrane, deliver the cell, deliver the transgene or the genetic material. That genetic material pass the cytoplasm, approach the nucleus, enter into the nucleus, then attach with the DNA. When it is attached to the DNA, it becomes integral part of the DNA. When it is attached with the DNA, then it start its functioning. We talk, when we say it started functioning, it means it start producing the messenger RNA. When messenger RNA is produced, then messenger RNA travel all the way down to the ribosome. Then translation occurred. In transcription, messenger RNA produce, messenger RNA then travel all the way down to the ribosome. In ribosome, they are translated and the protein synthesized. If you say the protein may be the enzyme, maybe the hormones, it may be the modulator, maybe transmitters, maybe neurotransmitters, it may be the functional, it may be the structural. So our major goal is to restore the function of particular targeted tissues or the cells to obtain the proteins. Gene therapy, there are the two main methodologies used in gene therapy: in vivo gene therapy, ex vivo gene therapy. In vivo gene therapy is the delivery of the new genetic material directly into the targeted cell within the body. Ex vivo gene therapy, the targeted cell are removed from the body and then genetically modified. This is a safe method, but dependent on the type of the cell being targeted. There are the 7 steps involved in the process of the gene therapy: identification of the gene responsible of the disorder or the disease. In second phase, we make copies of the normal gene. In third step, the direct delivery of the gene into the host cell or the targeted cell. At the fourth stage, incorporation of the gene copied into the carrier virus vector. At the fifth stage, infect the affected cell with the virus vector. At the sixth stage, activation of the gene so that the transcription translation should occur. At the seventh stage, the desired functional structural protein synthesized or produced within the body. Okay, well, this is the diagram that actually highlights the delivery of the gene incorporated in adeno-associated virus into the targeted cell or the targeted organ, targeted tissue within the body. The cell to obtain the receptor or the secretory proteins, the DNA that is encapsulated in adeno-associated virus, then injected into the patient or the person or the individual. When it is injected into the individual, adeno-associated virus release gene into the cell. That cell start expression to produce the proteins into the targeted cell. The proteins may be the receptor proteins, but that may be the secretory proteins. Okay, well, in last conclusion, the gene therapy can potentially be used for the treatment of the genetic diseases. The emerging technology can possibly be used to unveil the fact and explore the hidden information that will help to rationalize the new method of the gene editing technology. Okay, well, these are my colleagues. These are my supporters, well-wishers. I would like to salute it and greet these friends, including my son, Sherry Bajkat, Wolfgang, Daniel, Terry Mill, Robert Heyman, Aslan Taha, he's my son, and then Kunza and Noreen. I acknowledge this presentation today to the Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery Technology Development Incorporation Canada. Both of these organization institutions located in Ontario, Canada. I appreciate your time and watching my video today. Thank you very much. See you soon. Take care.